Now we're on to inhibitors of oxidative phosphorylation. Rotenone is going to inhibit complex 1. This actually also helps the natives of the Amazon rainforest to catch fish. What they actually do is beat the roots of certain trees along the riverbanks, and those trees will actually release the rotenone, which then will paralyze the fish. And then paralyzed fish are a lot easier to catch than the ones that move around. Cyanide, azide, and carbon monoxide all inhibit complex 4. What they do is they bind tightly to the ferric form of one of the cytochromes there. Oligomycin is an ATP synthase inhibitor. So this basically gives you an overview of the different ways to inhibit oxidative phosphorylation. So what you'll notice is that there's actually 4 main places of inhibition, that of the ATP synthase, complex 1, and complex 4. There's also then a 4th one that actually is just pointing to the membrane and lists a set of uncouplers. We'll talk more about uncouplers here towards the end of the video. But what you should notice is that any of these things will inhibit oxidative phosphorylation. Some of these things are actually inhibiting the production of the proton gradient. Others of these are then inhibiting the actual ATP synthase itself. Finally, uncouplers are also going to affect the proton gradient. The interesting thing, though, is that oxidative phosphorylation is not the only way the cell can make ATP. But why are these inhibitors so toxic? Go ahead and give yourself some time to think about this one. Now, something else you may have noticed in the creation of ATP is that all the ATP that we've made is now in the matrix of the mitochondria, which is then needed by the rest of the cell. So we have to have some way to get the ATP out. There is a translocase that is then going to go between the matrix of the mitochondria and the cytosol and will allow the ATP out of the mitochondria. So since you have the ATP with a -4 charge moving out of the matrix and an ADP with -3 charge moving into the matrix, you have a disruption in the balance of the charges there. So since you're actually losing something that is more negative, the ATP, and gaining something that's less negative, the ADP, this is essentially as if we had transported a proton into the inner mitochondrial matrix. Now notice we're not actually transporting a proton into the inner mitochondrial matrix. What we are just saying is that because of the difference in charges, the matrix then becomes less negative overall. And that will also have an effect on our proton gradient. This is just to show you that the ATP-ADP translocase in the mitochondrial membrane has multiple intramembrane helices. Looking at our P/O ratio for mitochondrial electron transport and oxidative phosphorylation, is trying to look at how many ATP can you make per electron pair sent through the chain. In order to think about this, we need to consider how many electrons we're going to get per every part of the electron transport chain. So if you'll remember, when we're looking at NADH, we get about 10 protons pumped. When we're looking at our FAD in complex 2, we get about 6 protons pumped. So starting to look at this, we need that notion of how many protons do we get per electron pair out of our electron transport chain. As far as the ATPase is concerned, that's also going to depend on how many subunits it has. So we're going to assume that there's 10 C subunits. If you'll remember back from that chart, there's actually multiple numbers that can be in that chart, anywhere from 8 to 15. But for the sake of this, 10 is a very common number, so we're going to assume that there's 10 C subunits. With 10 C subunits, it therefore requires 10 protons to drive one complete turn of the enzyme. One complete turn will generate 3 molecules of ATP. So here what we're looking at is the proton to ATP ratio is about 10 to 3, or 3.33. That is, you get 1 molecule of ATP per 3.33 protons transported across by electron transport. So therefore, if you make 1 ATP per 3.33 hydrogens, the number that you then make per 2 electrons is about 10 divided by 3.33. So notice that we're getting the fact that we get 10 protons for every 2 electrons. So here, that 10, we're actually using the number of NADH that we get. But 1 proton is going to be consumed in the export of every ATP molecule. So a better ratio is going to be using that 10 divided by 3.33 plus 1. That plus 1 then accounts for the cost of ATP transport. So therefore you get about 2.3 ATP molecules. Notice that if you had an ATPase with 8 C subunits, a smaller number of C subunits, you get a different number out the other end, at about 2.7 molecules of ATP. Also, in bacteria, there's no need to transport ATP since there's no organelles, there's no mitochondria. So without the mitochondria to worry about, those P/O ratios are even higher. But how are the electrons of the cytosolic NADH fed into electron transport? So NADH doesn't actually cross the inner mitochondrial membrane. So therefore, we actually use shuttles to affect electron movement without actually carrying NADH. There's two main ways that do this. One is in the Glycerophosphate Shuttle, which will store electrons in glycerol 3-phosphate, which can transfer the electrons to FAD. The other is the Malate-Aspartate Shuttle that's going to use malate to carry the electrons across the membrane. So here what you see is our dihydroxyacetone phosphate going ahead and then being reduced to glyceraldehyde 3-phosphate. That then takes an NADH molecule to do. From there, the glyceraldehyde 3-phosphate can then be transported into the mitochondria, where then it can go ahead and where it can then be utilized by a protein inside the mitochondria to then do a mitochondrial reduction of FAD. The other option is the malate-aspartate shuttle. The malate-aspartate shuttle connects to a lot of other pathways, but what you'll notice here is that in the cytosol, you can go ahead and reduce oxaloacetate to malate. That then will require the change of NADH to NAD. So we're using our NADH reducing power to go ahead and reduce malate. Malate then therefore has the extra electrons. Malate can then move from the cytosol to the mitochondria by using this malate carrier. That will then essentially exchange a malate for alpha-ketoglutarate. Once the malate is in the mitochondria, it can then be oxidized back to oxaloacetate, which will then reduce NAD+ back to NADH. And then the NADH can be used in the electron transport chain. The oxaloacetate can actually go ahead and leave again by having the aminotransferase go ahead and take the amine group off of glutamate to go to aspartate. The aspartate can then go back out into the cytosol. The leftover glutamate, once the amino group is gone, is actually alpha-ketoglutarate, which can also go back into the cytosol. So what you'll notice is that between a malate dehydrogenase and an aspartate aminotransferase, this allows this process to go however many times it needs to with a relatively few number of carbons, all to transport electrons into the mitochondria. So here's a look at the big picture. Looking at the citric acid cycle overall, we get 3 NADH, 1 FADH2, and 1 ATP generated. Notice that this is starting with acetyl-CoA as a beginning point. Then if we're going to look at the complete oxidation of glucose by both glycolysis and the TCA cycle, now we're starting with glucose as a beginning substrate, and we wind up with much larger numbers, that of 10 NADH, 2 FADH2, and 4 ATP. Those 10 NADH and 2 FADH2 make about 34 molecules of ATP in electron transport and oxidative phosphorylation processes. But the question is, as always, how do we get here? So when we're looking at this, we're going to use those P/O ratios to figure out how we got here. So we're still assuming that there's 10 C subunits per F0 rotor. So for this, if we're getting 10 protons per NADH, and it requires 10 protons per 3 ATP molecules. We're also going to assume that we get 6 protons per FADH2, and again, still 10 protons to make 3 ATP. So therefore, we get about 3 ATP molecules per NADH, about 1.8 ATP per FADH2. Since there's 10 NADH, that'll get us 30, and 2 FADH2 will get us another about 3.6. That rounds up fairly well to 34. Now notice that this 34 is going to be if the malate-aspartate shuttle is used. The glyceraldehyde 3-phosphate, if you'll remember, is actually going to use FAD instead, so you actually get a couple fewer ATP molecules if you're using the glyceraldehyde 3-phosphate shuttle. But what about uncouplers? Actually save this one for last because it's one of my favorites. So uncouplers will actually disrupt the coupling of electron transport and the ATP synthase. These are relatively small molecules, but what they are is they're hydrophobic molecules that have a proton on them. What they can actually do is move through the membrane without any sort of shuttle, without any sort of other proteins involved. They'll just flit through the membrane itself. And what they can then do is they can carry protons across the membrane. Since this is just a chemical process, they're going to carry protons across the membrane in the direction of the gradient, which means they're going to basically destroy the gradient over time. They will cause the proton gradient to dissipate. Now, this sounds terrible, right? But uncoupling the proton gradient produces heat. When might producing heat be useful to an organism? I'm going to go ahead and tell you, because again, this is actually one of my favorites. This is actually how bears stay warm in the wintertime. This is also how newborns help to regulate their body temperature. So this notion of uncoupling and having this proton gradient be a little bit leaky, being a little bit leaky is actually useful because uncoupling that proton gradient does actually give heat, and heats can be really valuable under the right circumstances.